Hi, Joseph. It's great to have you here. I understand that you're working on breast cancer, and I'm wondering if you could tell us a little bit more about exactly what you're trying to figure out.
So something our lab is really fascinated by is a process called collective invasion. Essentially, it's cancer cells actually moving together in packs of cells rather than single cells. And that's one area that we've been really fascinated by because we didn't envision that cancer cells would actually cooperate to move. And so that's one process that currently we're trying to understand further. And not just that, but we're trying to also understand why tumors actually have a lot of this diversity in the first place. Some cells do actually cooperate together when they move, and some cells actually move as single cells. So trying to understand what allows these cells to move in either way is definitely something we're interested in.
So it sounds like tumor cells don't always invade as a group. Sometimes they do. But if it's advantageous to invade as a group, then how come it doesn't always happen?
Yeah, we would envision that if it is really advantageous that cancer cells would move in groups, that they would do it in that way. And that's actually one thing that we're really intrigued by. We actually find that single cells, we always find in tumors that single cells do invade out kind of on their own as well. And we've always been curious as to why that's even present in the first place. And I think that's just because tumor cells, for the most part, they are very heterogeneous. And what we mean by that is that there's a lot of diversity. Even among breast cancer, there's a lot of different subtypes. But we've also found that in a single tumor, there's also a lot of different groups of cells that do different things. And so I really do like to think of a tumor as kind of like a company, where in some ways we have different, even within a company, there are some departments or some groups that over time maybe lose their efficacy or they lose their function. They're not really as useful anymore. And you'd imagine that they kind of phase out. And so in the same way, cells are very similar in that through evolution, some groups of cells you would imagine if they're not really as needed, they would not survive. They would actually kind of phase out. And so that's what we would imagine for single cells if they're not as needed within a group, within a tumor, but we actually find that it's still there. And so there must be a function that these cells have that is necessary for this tumor to grow and to invade, and we're trying to understand why that's the case.
It seems like systems biology is a great way to study biological diversity that you were talking about. And so I'm wondering if you could tell us a little bit about what types of tools and approaches that stem in systems biology you're using to understand this sort of single cell versus collective cancer cell migration.
So one technique that our lab actually developed is to actually take a single tumor and be able to actually separate out different groups or different subpopulations, we like to call it, of cells that act differently. And so something that we've already done is we've taken a single tumor, we've seen how they invade, and we've noticed again that there are some cells that move in packs of cells or through collective invasion, and some cells that elect not to move in that way. And so we've actually used our technique to be able to separate those cell populations out. And what we've done is we've done gene expression analysis. And so what that means is we've actually looked to see how the gene expression of these different groups of cells are different. And we've been trying to basically compare them to see basically how they are different. And potentially using that information, we're trying to understand what roles each of these subpopulations could be playing for the whole tumor to eventually grow and to invade and metastasize to a secondary site. So that's something that we've been trying to do, looking at these differences and seeing what roles these cells might play.
And have you found anything surprising?
I think the most surprising thing that we've been able to find is that when we compare cells that invade cooperatively versus cells that don't, we found that cells that invade cooperatively, actually, they produce a lot of cholesterol. So that's really surprising to us because for the most part in humans, it's mostly liver cells that produce cholesterol. But we're actually finding that breast cancer cells that invade cooperatively, they actually produce a good amount of cholesterol. And this is interesting because there are actually cholesterol reducing agents that are FDA approved called statins that have been used in breast cancer treatment in the past, but their efficacy has always been up and down. Sometimes it works, sometimes it doesn't, and it's been very inconsistent. And maybe this illuminates a little bit of why that might be the case because of the fact that tumors maybe they are very diverse, and maybe the fact that there are some tumors that will invade cooperatively more versus tumors that invade a single cells more, that could be a reason why some statins could work maybe for cooperatively invading tumors, but not so much the ones that do not cooperate. And so that's one thing that we're thinking about right now and trying to figure out whether or not that correlation is there and whether or not that's one reason why statins might actually work for breast cancer treatment, but only certain types of breast cancer treatment.
Interesting. I guess we'll have to keep our eyes open for that, hear what happens in the future. So as I listen to you describe your research, one of the things I find really fascinating is that you are straddling sort of the line between basic research, which really is about how the cells work, how they communicate to one another, understanding those types of things in the context of breast cancer. And then also translational research, which is a lot more about sort of what have we learned and what can we then take to the bedside, in terms of novel therapeutics or diagnostic tools that can help patients. What has motivated you to pursue this type of research?
Intellectually, I think the idea of tumor diversity, or the fact that there's in some ways kind of like a community of cells that work together to form a tumor, I think the complexity of that was really fascinating to me. And so that's definitely something that motivates me. But also working in a hospital setting, I think really reminds me of how real this disease is. We know that for breast cancer patients, one of the main things that leads to death is the fact that the breast cancer tumor actually does invade and spread to a secondary site. And so trying to find a way to stop this process or to mitigate this process is definitely something that also fuels me to try to understand what the differences are in the different groups of cells that are present and try to kind of stop this process from happening in the first place.
Well, thanks so much for sharing your work with us.
Of course, my pleasure.